Welcome to today's live broadcast. Targeted single-cell RNA sequencing enables genome-scale CRISPR screens. I am Jen Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Illumina and BD Biosciences. For more information about our sponsors, please visit illumina.com and bdbiosciences.com. Now let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click the Send button. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. I'd like to now introduce our presenters, Dr. Keilholz, Dr. Zajac, and Dr. Buerckstuemmer. For a complete biography on our presenters, please visit the Biography tab at the top of your screen. Dr. Keilholz, you may now begin your presentation.
Thanks a lot, Jen, for the kind introduction, and it is my honor now to start this webinar today on CRISPR screens. And I would like to briefly introduce the BD Rhapsody single-cell analysis system powering biological research. I hope you can all see my screen okay. And with this, I would like to go to the first slide with a little bit of an introduction on BD Biosciences. So BD Biosciences is active and is supporting biological research on the single-cell level for over 40 years now. With the invention of the FACS technology and the first FACS cytometers coming to life in 1974. Since then, we have developed as a company, as the market leader really in flow cytometry, with nowadays over 9,000 instruments installed just across Europe and over 92% of citations in flow cytometry instrument citations. We now, being a market leader is nice, but the challenge really stays in staying a market leader, and this requires what we are actually doing. We are having an increasing portfolio of reagents and instruments and keep on further developing this. For now, we have more than 20,000 flow cytometry reagents on our catalog. We have the BD Horizon Brilliant dyes. We have the BD OptiBuild on demand portfolio, custom reagents, and the BD OneFlow solution for diagnostics testing as well. That's on the reagent side. Certainly, BD keeps investing in innovative instrumentations, and we have a whole portfolio of instruments and ever keep increasing the platforms here. And one platform I would like to talk about today very briefly, which is the BD Rhapsody system. However, as all of BD Biosciences, we really want to be and stay your partner of choice when it comes to scientific solutions. And this is supported by powerful and intuitive software solutions and tools that we are offering. But last not least, our very skilled teams of field applications support who are helping you in your labs with your biological research. Now I would like to briefly introduce the BD Rhapsody system to you and just show the workflow components here. On the left-hand side, you see the BD Rhapsody scanner. Below the scanner, you see an electronic pipette, which belongs to the system. And then we also have the BD Rhapsody Express, the cartridge station. Now, the core of this technology really is the BD Rhapsody cartridge, which you can see on the right-hand side. And in order to explain a little bit more what is happening here, I will advance to the next slide. On the left-hand side, you see the microwell cartridge, and this is actually already saying what it is. It is a cartridge made of microwells. And as you can see in this little insert, these hexagonal shape of microwells, we have more than 200,000 of those on the cartridge. And the idea is to load the cell solution or the cell dispersion into your cartridge, let the cells settle down into the microwells, and individualize them by this process. All of this in the lab is supported by the BD Rhapsody Express station that you can see in the middle here. And this is actually a very small tool, a very not complicated tool. It doesn't even need electrical power. You can easily put it under the hood and manipulate the cartridge with the help of this little device. Below here, you see the workflow, what is happening on the cartridge. The idea is you load your cells and the beads. And you can see in this little insert down here, there is one of these hexagonal shaped microwells sitting in red, or in orange, there's a bead sitting in there, and in blue, there is a cell sitting in there. That's the idea of the system, coupling cells in one cell with one bead. This is how you achieve the resolution on the single cell. Next step in this workflow, you would lyse the cells. As you can see, hopefully here, the cell in blue is sort of melting away, and the yellow, no, not yellow, the orange bead now has a coating in blue, which reflects the analyte molecules that are captured by this bead, which is mRNA and potentially also tags from antibodies. I will come to this a little later. What also happens then is you will retrieve the beads. The beads are magnetic, and a magnet is applied from the top of the cartridge, and this is happening in the express station. And then collect all the beads into a single vial or into a single Eppendorf tube, and where you then do the downstream chemistry in the lab. So you synthesize the DNA and the cDNA, and after creating a library for next-gen sequencing, in the end, then you analyze the data. Now, what is the scanner for? The scanner is an imaging tool, and it is used in quality control. As you can see on the top of this slide here, this is the workflow. You start with the cell count, cell variability check, the cell, the bead capture, lysis, bead retrieval, then library prep, as I said before, and on the right-hand side, we we go to an Illumina system and do the next-gen sequencing readout. All of this experiment is valuable and costly, and we want to get as much control over it as possible. And this is where we use the scanner for. We use the scanner actually already for cell count and cell variability testing, which is not seen on this slide here, but I put down the steps, cell bead and bead capture, and the lysis and bead retrieval. And as you can see, here are some real images on the left-hand side of these images. You see a cell sitting in the microwells, and I've circled them in orange for better visibility. So these are individual cells. And you see that this cartridge is heavily underloaded. The reason for this is we want individual cells to be sitting in there and not multiplets, that is 2 or more cells per well. In the middle, we have applied the beads, and as you see, we overload the cartridge. Now we have many more beads in there than needed, but the idea is to have 1 bead per microwell. There is no space for 2 beads to be put in there, so we are happy and we are safe on this side. What I do not have here is we wash away the excess beads. This is a picture which we also acquire by the scanner, which I just simply don't show here. On the right-hand side, then we see after retrieval of the beads, we want to make sure that the cartridge indeed is empty. And as you can see, this is the case. So by using the scanner, you have a full control over the entire workflow. And that means you are not running like in a black box. Certainly, we really want to be nice to ourselves. This is about transcriptome assessment and transcriptome analysis. And you all know that cells keep adjusting their behavior based on what they see from the outside. So in other words, if you apply stress to a cell, the cell will change its transcriptome towards stress-related genes and express them more strongly. This certainly is what we don't want. And please keep in mind that all of us, all of our cells, all cells on Earth, for their whole lifespan, are exposed to gravity. So gravity is something that is really normal to every single cell. The buffer system that we are using the cells, or that we put the cells in, is a physiological buffer. So cells in a physiological buffer are most likely quite happy. And then we are using gravity to individualize them. As I said before, the cells just slowly sink into the microworlds without putting any pressure on them. So this is as physiological as we can be. And as you can see here down in the graph, what we did, we compared the Rhapsody outcome to flow cytometry, which is still seen as a gold standard. The orange bars are flow cytometry data. On the x-axis, we have different cell types of a composition of cells. On the y-axis, we have the percent of live cells in that samples, and this is based on 2 different replicates from 2 different mice. And as you can see, very roughly, flow data and Rhapsody run number 1 in blue and Rhapsody run number 2 in gray is pretty much in the same range. So this proves that we really also on the Rhapsody see what we would see on a flow instrument. And on the BD Rhapsody, we can currently load and process about 40,000 cells in one go on one cartridge. So this is what we currently have to offer. On the left-hand side, the instrument, of course, and we can address cells and nuclei. In the middle, you see the assays that we are currently offering, which is a whole transcriptome amplification assay, I should say 3' biased. A targeted RNA-seq assay, which we are going to hear a lot more about today. And the sample multiplexing kit, VDJ immune repertoire testing, and AppSeq. I'm not going to talk about these today. On the right-hand side, all of this is supported by bioinformatics tools that make your life hopefully easier because they should be easy to use. Now, this is an animated slide, and this is almost my final slide here. So the BD1 solution really is, we have a discovery workflow starting from the left-hand side here with blood and tissue or cell culture that you can put into the BD Rhapsody system. You have a heterogeneous cell population here. You can apply sorting for enrichment by using a FACS sorter. Then, of course, the discovery part comes where the BD Rhapsody is really positioned. And then the computational analysis, which is also known as the primary analysis, and then certainly on the informatics side. As I said before, BD has some solutions to help you with with generating or visualizing your data. And with this, I'm coming to the end of my presentation, of my short presentation today on the BD Rhapsody system, and I will be more than happy to hand over now to Pawel from Illumina. Thanks a lot.
All right. So thank you very much, Wieland, for this really nice introduction about single-cell sequencing and the BD Rhapsody system. And of course, thank you very much to all of you for joining us at this exciting webinar. And as mentioned, my name is Pawel Zajac. I'm part of Illumina's marketing team, and it's my great pleasure to, in the next 5 minutes or so, present Illumina to you, who we are, what we do, and also tell you a little bit about our latest sequencing news. So for those of you who haven't heard about Illumina, we are a global company, and we develop, manufacture, and market innovative next-generation sequencing and microarray technologies. And our technology enables you to make foundational discoveries to transform medicine and ultimately to impact lives. And to date, there are more than 200,000 publications where our technology has been used, so it clearly shows that this is a robust technology that you can trust. And there are 2 ways in which you can get access to our technology. You can either purchase a system and implement it in-house in your own lab, but you can also turn to one of the many service providers and get access to our technology through these service providers. So we do provide next-generation sequencing systems. And it is a single technology, but it unlocks a range of applications and a range of biological insights. So the sequencer can be seen as a tool, and by preparing the sample in different ways, you can use this tool, the sequencer, to get access to a multitude of biological insights. So if you take a look at, for example, cancer research, of course, you can use our sequencing systems to do single-cell sequencing. But you can also perform tumor mutation profiling, for instance, via whole genome sequencing or exome sequencing, to figure out what differentiates the tumor from normal tissue. And you can also do, for example, 16S sequencing or shotgun metagenomics to figure out what is the microbial component and how does the microbial component affect the tumor. And our sequencing systems, they enable a wide range of single-cell applications. So no matter which biological modality you're interested in, whether that's RNA or DNA or protein or different regulatory features, and no matter if you want to do a global analysis or you want to zoom in on a targeted set of, for example, transcripts, we do have sequencing solutions that fit your output needs. And it's exactly the same with your study size. So irrespective of if you have a small study or a large study, we are more than happy to assist you with the sequencing part. And we have a nice portfolio of sequencing systems. At the bottom left-hand side of this slide, you can see our iSeq 100 system. That's our smallest, most affordable benchtop system, giving you 4 million reads. And on the right-hand side, we do have our NovaSeq 6000 system, which is our production scale sequencer for those larger studies. And earlier this year, we launched our NextSeq 2000 platform, which allows for deep and large studies, but still within a benchtop package. So there are a number of technological innovations that have gone into making the NextSeq 2000. And it clearly shows our long-standing tradition of technological innovation. And all these technological innovations, they translate into a number of significant advantages. So for example, there is a reduction in operating costs and the reagent kit size compared to the NextSeq 550. And also the output is increased from 400 million reads that you get with the NextSeq 550 to 1 billion reads that you get with the NextSeq 2000. So paving the way for those larger and deeper and broader single-cell sequencing studies. And just a couple of months ago, we also released the latest version of the consumables for our NovaSeq 6000 system, so our production scale sequencer. And these consumables carry a number of advantages as well. And one is a attractive price point, meaning that you can sequence more within the same sequencing budget. So this once again opens the door for those larger, broader, and deeper single-cell sequencing studies, allowing you to see more of what's happening in your biological samples. In addition to the price change, the new consumables also have a number of additional advantages, such as greater flexibility, improved robustness, and enhanced user experience. To name one example, the shelf life has been increased from 3 months to 6 months. And if you have any questions about any of our solutions, maybe you are considering a project and you're interested in discussing different options, please don't hesitate to reach out. We are more than happy to assist. So with this, I very much wanted to thank you for your attention. And I will now turn the presentation to Tilmann, the star of today. Please take it away, Tilmann.
Excellent. Thank you, Pawel. Just a moment. Okay. Can you see my slide? So, thanks again for the very nice introduction into the BD Rhapsody system, Wieland, and thanks for introducing us to the Illumina product portfolio. My name is Tilmann Buerckstuemmer, and I'm the Chief Scientist of Alien Biotechnology based in Vienna in Austria. And the title of my talk today is Targeted Single-Cell RNA Sequencing Enables Genome-Scale CRISPR Screens. Now I have to quickly show this disclaimer slide before going into the introductory slide. So at Alien, we run CRISPR screens. CRISPR screens rely on the CRISPR-Cas9 system, which can be used to disrupt any gene of interest. And the main application of the technology to date, at least with clients in industry, is target discovery and target validation. So a lot of the drug discovery programs these days start with a CRISPR screen. And so the way a CRISPR screen works is we start with a population of cells in which we introduce the Cas9 endonuclease together with a library of guide RNAs. And now this library of guide RNAs will establish a population of gene knockouts. And then we have two screening paradigms. One is the positive selection screen, where some clones, like the ones shown here in red, become more dominant. On the other hand, we have a negative selection scheme where certain clones disappear in the experiment. Now, what are the readouts that are currently used for CRISPR screens? The simplest readout that you can think of is a fitness screen where, for instance, you might start off with this population of gene knockouts and treat cells with a drug. And in the course of the experiment, some of the gene knockouts, as a consequence of the drug treatment, will become more or less dominant. The advantage of those screens is that they're very scalable. People have done them, in fact, at genome scale. But in an ideal world, you would like to be able to address more complex biological questions. And so there is a nice middle ground here, which is the CRISPR screens that are based on flow cytometry. They exploit cell surface markers, which we can stain with fluorescent antibodies or intracellular reporters. But what you would really like to be able to do is perturb a set of genes in a given cell and then read out essentially all the changes as they happen as a consequence of the gene knockout. So this is a multi-parametric screen. But of course, that is sort of a more difficult undertaking. And we think single-cell RNA sequencing is a very interesting approach for those types of screens. So at Alien, our typical workflow starts off with a Cas9 expressing cell line. So we stably introduce Cas9 into a target cell of our liking. We then perturb a set of genes in a pooled fashion so that within a single Petri dish, we have knockouts for different genes growing side by side in the same dish. And then we're singling them out and creating a single-cell RNA sequencing data set. I'll speak about the technicalities of that in a moment. But effectively, we're recording the transcriptome of any single cell in that Petri dish, and we're mapping it back on the guide RNA that was causing this perturbation. And obviously, because we're dealing with large data sets, we have a need for a computational analysis, which is depicted here on the right side of the slide. Now, the difficulty in joining CRISPR screens with single-cell RNA sequencing was really to read the guide RNA that was expressed in a given cell. And that's difficult because most single-cell RNA sequencing platforms use beads coupled to oligo-DT for reverse transcription of the transcripts of a cell. And on the other hand, most CRISPR screens use a guide RNA expression cassette in which guide RNAs are not polyadenylated. And so there was a trick that was actually necessary to sort of make this happen that was developed by Pawel Zajac in the lab of the co-founder of Alien, Chris Bock. And the trick was effectively to put this expression cassette within the context of a polymerase II transcript so that one could create an indicator transcript that can be captured with oligo-DT coupled beads. So if this is sort of the sequencing platform, how do we start? Typically, we start these CRISPR screens by designing a set of guides. And for any CRISPR screen, it's very important to design the best possible guides. And so we've developed a workflow in-house that not only incorporates the sequence information, the local sequence information, but actually also takes other features of the sort of local DNA sequence into consideration, most importantly, evolutionary conservation, but also domains of the proteins that are encoded by this specific region and some additional features. And we think it's very important because, again, the guides or the effectiveness of the guides that are used in the screen ultimately determine the signal-to-noise ratio in the experiment. So then as a next step, once sort of guides have been designed in the computer, if you like, we need to clone them and create a lentiviral guide RNA library. And the quality that's typically done for such a library is shown here. So typically we take either the plasmid library or the lentiviral library, and we sequence the guides that are present in that library. And for a CRISPR screen to be unbiased, it's very important that we have a very equal distribution of guides across the entire library. And we feel that the approach that we developed here warrants that, and warrants that very few guides drop out. This is a relatively big library that we're showing here of 11,000 guides, and we're just losing 21 guides. Or I should say only 21 guides were represented at less than 10% of the median. So we think we have a very homogeneous way of cloning these libraries. So what are the quality control steps that we apply to the cells that we work with? Now, there are two types of workflows that we typically look at prior to starting a screen. One is we take a Cas9 expressing cells and we inactivate a cell surface marker, and we often use CD81 for that. And so what you can appreciate is that cells that are untransduced, let's say the parental controls here, they have sort of 98% CD81 signal. But then the moment we're bringing in the guide, which also carries GFP, we're losing CD81 signal sort of greatly. The other thing we do is we're targeting essential genes, and that is shown here. So two examples are KIF11 and PLK1. So we're targeting these with guide RNAs, disrupting these essential genes. And as a consequence of that, cells will die in the course of the experiment, which we're visualizing here because the cells are pre-labeled with GFP. And again, what you can nicely appreciate is that such an experiment allows us to determine the time point at which we conduct a CRISPR screen. And in this particular example, we picked day 8 for the experiment. Now, the experiment we focus on, and this has become a little bit of a workhorse for us, is focused on the signaling pathway that is elicited by interferon beta. And the reason is that the signaling pathway triggers a very defined transcriptional output, which we can capture with single-cell RNA sequencing. But also the signaling pathway is one that has been studied for at least 2 decades. And so we know the signaling mediators in that pathway very well. And so what we did then is we targeted those mediators that we know from the literature play a role. And we supplemented the library with additional sort of friends and family, and then conducted the CRISPR screen. And so the workflow, again, is depicted here. We're starting off with a guide RNA design process that I've explained to you on the previous slide. We're then cloning the guide RNA library. In this particular example, we used 3 guide RNAs per gene. A few non-targeting guides were also included as reference points. We then make a pooled lentiviral library infect a Cas9 expressing cell and treated those cells with type 1 interferon or left cells untreated, and then prepped 2 independent libraries using conventional droplet-based sequencing. And when we sequence this experiment, these experiments get quite big. An unbiased experiment of about 10,000 cells is, roughly speaking, 300 million reads. And obviously, down the line, you need to do a computational analysis to make sense of that experiment. So how can we visualize that data? Of course, the data is relatively complex. We're dealing with about 10,000 cells in each experiment. That's sort of the standard unit, if you like. And in each of these cells, we want to ideally monitor 20,000 human genes. So these are large matrices that nobody can really understand. And so there are 2 ways of sort of visualizing data. One is the so-called UMAP plots that most of the single-cell RNA sequencing community is quite familiar with. Here, every cell is a dot. Importantly, these experiments or these types of analyses are unbiased, so they really cluster cells by their difference in the transcriptome. And if cells cluster together, it means that transcriptomes are similar to one another. Now, for crop-seq screens, where we perturb cells with guides, we also tend to like what we call here the aggregated view. So with the aggregated view, we're essentially aggregating over all the cells that have a knockout for a given gene, and we're analyzing the transcriptomic changes in that subpopulation, if you like. And so that allows us to create more conventional heat maps, which can visualize the changes going on as a consequence of the CRISPR perturbation. So let's have a first look at the data. This is the experiment I described to you, the screen where we treated cells or left them untreated with type 1 interferon. And when we do this unbiased UMAP clustering, you can easily appreciate that there are 2 big clouds. And if we color them by their origin, you can easily appreciate that the control cells are very nicely separated from the interferon-stimulated cells. And that means that a lot of the variation that we observe in the experiment is attributable to the type 1 interferon stimulation. If we then ask what are the genes that are up or down regulated in control versus interferon beta, we recognize a lot of the genes that we are familiar with. In fact, a lot of the genes that are regulated start with an I because they were first described as interferon-regulated or interferon-inducible genes. And so that seems to say that we're sort of uncovering or we're sort of detecting relevant biology here. So then you wonder, what are the genes? So what do the CRISPR perturbations do in this particular experiment? And here we're turning to the aggregated view that I just described to you. So for most of the genes, for most of the mRNAs depicted here on the y-axis, they're low in unstimulated cells and they get upregulated in stimulated cells. But then if we perturb genes on the x-axis with CRISPR, you can nicely appreciate that there is sort of a small set on the left side that, despite the fact that they were stimulated, they look very similar to the unstimulated cells. Those are positive regulators of the pathway. And then there is a small set of negative regulators shown here towards the right of the heat map. If you compare the little schematic that I showed you initially to this heat map, you will easily appreciate that a lot of the genes, in fact, all of the genes we uncover here are the genes that you would like to uncover from sort of previous and historic knowledge. Importantly, we find the 2 receptor subunits, IFNAR1 and 2, the 2 kinases JAK1 and TYK2, and then this transcriptional complex consisting of STAT1, STAT2, and IRF9. So this would suggest that the workflow is sort of up and running, and it can really recapitulate sort of known biology. Now, in the next part, I would like to explain how we can couple crop-seq screens with targeted single-cell sequencing. And if we go back to this data set that I just showed you and we plot all the 20,000 human genes and we compare their expression level to the variance observed in the experiment, you will easily appreciate that most genes don't change much in in the experiment. So those are the ones boxed in black. And those genes are sort of not entirely ideal because they don't provide a great degree of information. And so what you would ideally like to be able to do is zoom in on those genes boxed in orange, which change between cells and which thus contain all the information that's relevant. So how can we do that? How can we zoom into those genes? This is where the BD Rhapsody machine became very handy for us. So the BD Rhapsody machine, as Wieland just explained, separates cells in a microwell-based approach. Then each well is labeled with a barcoded bead, and so on the surface of these barcoded beads, reverse transcription happens, much like we're used to sort of from more conventional approaches. But then the real trick here is the last step, the step number 3, where instead of amplifying all the RNAs of the cell, we're actually able to nominate a set of mRNAs that we would like to fish out, and we amplify those using a multiplex PCR. And this multiplex PCR is illustrated on the next slide. Again, reverse transcription happens on the surface of the beads. It transcribes all the RNAs of the cell. But then we can nominate a priori the mRNAs that we're most interested in, and we fish those out with a PCR-based step. Now, I would like to give you some metrics of what sort of typical experiments conducted here at Alien look like. So I plotted here 15 experiments, and as you can nicely appreciate, we typically load anywhere from 25 to 50,000 cells on one cartridge. This is not what BD recommends. BD says, typically they recommend 10,000 cells, I believe, per cartridge. But our cells are labeled with guide RNAs, and as a consequence of that, we have a very easy way of getting rid of the multiplets that we do observe and that are expected based on Poisson distribution. So that allows us to actually load a lot more cells. We have a very decent capture rate, which is around 85%, and then a very acceptable multiplet rate that is typically anywhere from 5 to 10%. Of course, as we're increasing the number of cells we load, we're also increasing the multiplet rate. So let's now go to a crop-seq screen, and let's see how the targeted sequencing technology behaves. Here we compared a whole transcriptome experiment to a targeted experiment. The targeted sequencing run was conducted on the BD Rhapsody system. We focused on 73 pre-selected mRNAs. We looked at about 10,000 cells per condition, and we sequenced those at about 15,000 reads per cell. The whole transcriptome experiment was based on droplet sequencing. We did not do any pre-selection, obviously, and again, we sequenced 10,000 cells, but because it's unbiased, we typically have to sequence at least at 30,000 reads per cell, sometimes even at 50,000 reads per cell. And so the experiment we then did is the exact same experiment I just described to you. So we're looking at the same guide RNA library. We're looking at cells stimulated with interferon beta, and we're looking at the outcome of this on the level of the two platforms. And so with the targeted sequencing, the first thing I asked our computational biologist to do is, let's take those 73 mRNAs and plot them on the x-axis, and let's just ask which fraction of all cells in the experiment contains reads for those 73 mRNAs. And as you can nicely appreciate, roughly 2/3 of those mRNAs were really well represented across the great majority of the cells. Now, importantly, this representation is maintained even if we're subsampling the experiment. So we started the experiment and we did the actual experiment with 16,000 reads per cell, but we can of course pretend that the experiment had less reads. And as you nicely appreciate, even with as little as about 3,000 reads per cell, we get a very nice representation of those 73 transcripts across all cells. So that's very important because it would suggest to us that we can get away with roughly 1/10 of the reads that we normally use for unbiased sequencing, and that means we can squeeze a lot more information into one experiment. The same thing is sort of illustrated on this particular slide. So here we're looking at 20 clones containing non-targeting guides. That's the x-axis. Each of the 20 clones is sort of plotted on the x-axis, and we're looking at genes represented in the targeted panel. And as you nicely appreciate with whole transcriptome sequencing, the data is very sparse. There are a few transcripts, those 4 on the bottom here, that are present across all cells, but in whole transcriptome sequencing, the great majority is sometimes there and sometimes not there, and there's nothing you can do about it. Now, with targeted sequencing, we really overcome this problem, and the great majority of the cells, maybe with the exception of MX2, expresses the great majority of the mRNAs we want to sequence. So targeted sequencing really alleviates the sparsity problem that we otherwise face in single-cell RNA sequencing. So how does this translate into a functional effect? Now, obviously, the ultimate goal of the experiment is to see which knockouts have transcriptomic phenotypes. And so we went back to this heat map that I showed you earlier, and we compared again whole transcriptome sequencing to targeted sequencing. But this time we adjusted the scale to the scale that we observed in the targeted sequencing experiment. And as you can nicely appreciate, first, the experiment is qualitatively similar, meaning the same genes have knockout phenotypes in both experiments. But targeted sequencing, despite the fact that it's read at 1/10 of the number of reads in this experiment, is a lot more sensitive. So the dynamic range of the experiment conducted with targeted sequencing as a workflow is much greater than with whole transcriptome sequencing. Finally, we asked how many cells do we need to get a meaningful phenotype? Again, this is an important question because most sequencing workflows have a fixed number of cells that they can swallow. And if we can have less cells per genotype, it means our screens can be enhanced in terms of scale. And so here we looked at a number of knockout marker pairs that were present in the screen I just described to you. And we plot the statistical significance as observed in the experiment against the number of cells that we need to get to that statistical significance. And so if you just sort of pick out one example, you will appreciate that whole transcriptome sequencing, or let's say globally whole transcriptome sequencing curves, are much more shallow than targeted sequencing curves. And if we just look at this one example here, let's say this marker pair of MX1 stat1, we need approximately 75 cells in the whole transcriptome approach, while we only need sort of 12 to 15 cells in the targeted approach. So this would suggest that we can not only use less reads, but we can also use less cells in the experiment and come out with the same level of statistical significance, which I think is great news. Now, in the last sort of 5 to 10 minutes, I would like to focus on a different data set. And sort of one of the questions that we first had when we started using the BD Rhapsody machine is, how sensitive is that machine and how sensitive is that workflow? And so in order to address this question, we actually started from a cancer cell line for which we had bulk RNA sequencing data. And so from that bulk RNA sequencing data set, we knew that we roughly have 12,000 genes expressed in that cell. And we then distributed these 12,000 genes into different boxes, if you like, or different tiers, and picked from the different tiers, as illustrated here, 35 genes per tier. And we asked, how good is the targeted single-cell sequencing workflow at detecting those genes? Now, we already know that with whole transcriptome sequencing, especially as you get to the sort of tier 1, 2, 3, it will be quite difficult to see these genes reliably. But the question is, can you bump them up? Can you bump them up with the targeted sequencing workflow that I just described to you? And this is the data as they came out. So here we're plotting whole transcriptome data against targeted sequencing data, and we're asking what's the percentage of cells in which the respective gene was detected. And you can nicely appreciate that if the gene is very abundant, like those pink genes up here, then both workflows would be sort of almost equally good at detecting the genes. But as we're going to less and less abundant genes, meaning to sort of blue and green, it gets essentially very quickly very difficult for whole transcriptome amplification to actually see those genes in a significant fraction of the cells. Whereas with targeted sequencing, it's actually remarkable that it can still pick these mRNAs up. So I think the only tier where targeted sequencing really starts to struggle a little bit is the tier 1 or 2, which is really very, very close to the to sort of unexpressed or not expressed genes in a cell. And a similar thing is illustrated on the right side of the graph, where effectively we're plotting the same thing across the 7 tiers for whole transcriptome amplification and targeted sequencing. So for the abundant genes, not a big difference, but especially for those intermediate and lowly expressed genes, targeted sequencing really makes a difference and really increases the coverage at which we detect those genes. So with that, I'm at the end. I would like to conclude that we're very excited about the targeted single-cell RNA sequencing workflow. It allows us to use a lot more cells per cartridge. I've shown you that we routinely do 20 to 30,000 cells per cartridge. We will need less reads per cell. We will need less cells per gene. But the great thing is that it sort of still comes at an improved dynamic range. So we really believe that this technology will pave the way towards genome scale crop-seq screens. So genome scale CRISPR screens coupled with a transcriptomic readout. I would like to thank my entire team here. The data presented today were mainly from Anke Loriger, Susan de Groof, Lukas Badacher, and Adam Krejci is our computational biologist. I would like to also acknowledge our scientific advisory board, Chris Bock and Jason Moffat from the University of Toronto. And I would like to finish with a slide that both Wieland, Pawel, and I have put together, which sort of contains a little bit of a guideline for those screens. So if your screen is a whole transcriptome screen and you want to run it with about 100 genes, typically those screens require 100 cells per gene, which means you end up having 10,000 cells in an experiment. You typically need about 50,000 reads per cell, so you need 500 million reads, and a good sequencing platform for that is the NextSeq 2000 or the NovaSeq S1. On the other hand, if you run a targeted screen, you might want to look at 500 genes. Keep in mind this is sort of already 5 times the number of genes I just described for the whole transcriptome workflow. You should be able to get away with less cells. We think 40 cells could be realistic, so that 20,000 cells would be a total if you multiply sort of 500 with 40. That would still be a single BD Rhapsody cartridge. 5,000 NGS reads per cell is what we envisage as meaningful for targeted sequencing experiments. And so that gets you to about 100 million reads, which is really one of the smaller NGS experiments, and the NextSeq 550 would be the right platform for that, the mid output. So I'm not going to go through all the numbers. I leave you with this slide. I'd be happy now to take any questions you might have on that slide or on any other slide. I would also like to again thank my co-speakers, Wieland and Pawel. We've had a lot of fun preparing this, and we hope you enjoyed the webinar so far.
Thank you, Dr. Keilholz, Dr. Zajac, and Dr. Buerckstuemmer for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is, what is the maximum number of sgRNAs you would suggest to add to your multiplex CRISPR screen without compromising editing efficiency?
That's a good question. I would say that it's, let me think about the question. I would say that typically, just to sort of maybe clarify, typically we deliver these guide RNAs at a low multiplicity of infection so that every cell harbors a single guide RNA. So we're not multiplexing in the sense that we're throwing all the guides into one cell or several guides into one cell. So if that is sort of the screening paradigm, it sort of is effectively one guide RNA per cell. The only question is, how many guide RNAs do you need to get to a meaningful biological outcome? And here I would say, again, it's quite dependent on the quality of your guides. There are some libraries that people believe they've curated very well, and they think they can get away with as little as 2 guide RNAs per gene. And some of the earlier libraries that were used in the sort of early 2015, 2016 had up to 10 guides per gene, or sometimes even more guides. So these days, I would say with a well-designed library, you should be able to get away with 3 or 4 guides per gene. I hope that answered the question.
Okay, thank you so much. We have another question. Is the Alien sgRNA Designer available as a service?
Yeah, I would say that this is totally something we can discuss. Typically, we don't do it as a standalone. So we sort of our business model is effectively to work with clients where we do end-to-end projects. So we're starting from sort of taking cells in and then all the way to the computational analysis. But I think we're small and we're happy to consider sort of any project.
Okay, thank you. I have another question. How do the number of detected genes vary among different cell types by using WTA and BD Rhapsody system?
That's a good question. I would say that from our experience, the main driver or the main barrier, if you like, for both platforms is the expression level, meaning with the abundant genes, they will likely be quite easily detectable with both workflows. But as genes get less and less abundant, the targeted sequencing workflow is really advantageous because it can bump up the efficiency at which you can detect intermediately or lowly expressed genes. And so if the question is sort of how well does it work across cell types, the question should be probably rephrased in how similar are those cells in terms of their transcriptomic profile, because that is sort of the key determinant. The other key determinant, and I didn't spend much time on that in the seminar, is that you need to have a good understanding of where transcription stops, because you're amplifying with a primer the 3' end of a gene. And if your 3' end is very poorly or not at all annotated, and you're several kilobases away from where you think that gene ends, it's very difficult to pick it up by a PCR-based approach. So you need to have a good bulk RNA sequencing data set to start with, to sort of maximize your chances of picking up all the genes that you would like to pick up.
Okay, thank you. I have another question. There are a few cases where targeted sequencing delivers less reads per cell than unbiased sequencing. What is the reason for this?
Well, the reason for this is that it's still a PCR-based approach. So one of the reasons is the reason that I just alluded to, which is you may not have annotated your transcriptional termination signal correctly. But this you can largely overcome by performing bulk RNA sequencing prior to designing your primers. The other possibility is that the algorithm, your primer design algorithm, fails. We've seen a few cases of that in the data, and you may have noticed that there were very few dots of the roughly sort of 220 genes that we amplified, where whole transcriptome sequencing was more successful in picking up the gene than targeted sequencing. There are a few such cases, but we haven't encountered many. So that there's definitely, I would say, less than 5% of all the genes we target seem to be difficult to sort of pick up because of issues with the primer design.
All right, thank you. I'm going to move on to another question. How long does the workflow on the Rhapsody take?
Yeah, I guess that question can probably go to me, so I'm going to answer this one. Remember, we want to be nice to ourselves, so we want to be to apply the least bias to ourselves and not give them a lot of time to react by transcriptomic change upon the experimental or the workflow conditions. So what I'm saying here is we keep them in physiological buffer. We just let them sit on the on this cartridge and settle them down by gravity. This certainly takes some time. So the typical incubation time on the cartridge is 15 minutes. And then once the cells are settled in there, they are the beads are added to it, which takes about another, let's say, 3 to 5 minutes. Which adds up to 20, and then we still do the scanning, which also adds a few minutes to it. So in roughly, I would say between lysis, between loading of the cartridge and lysis, we are looking at something like 25 to 30 minutes. And then once the cells are lysed, obviously the mRNA, the transcriptome, is captured by the beads, and nothing changes anymore.
Okay, I have time for one more question, and I want to remind our audience that those questions we were unable to answer today and those that come in during the on-demand period will be answered via the email address you provided at the time of registration. Our last question, I am interested in protein analysis, and you spoke about RNA-seq. How many proteins can you analyze simultaneously?
Yeah, I guess that question I will happily take on again. RNA-seq, yes. RNA-seq is interrogating the surface markers of the cells that are being expressed on the cells. And other than in flow cytometry, where the detection goes via light, so we are relying on the emission spectrum of the dye. And hence we are sort of limited in terms of mixing too many dyes to each other with each other, because the emission spectra at some point will overlap and hamper the analysis. This is not the case when you design, when you're able to couple oligonucleotides to your antibodies, and you can design them bioinformatically before you do so. So long answer short, we support panels as complex as 100-plex, and I do know of customers who do more than that, which at the moment is not officially supported, but we know it works.
Well, thank you again, Dr. Keilholz, Dr. Zajac, and Dr. Buerckstuemmer. Do you have any final comments for our audience?
Yeah, go ahead, Tilmann.
I just want to reemphasize that I think this is, at my point of view, the targeted sequencing workflow on the BD Rhapsody is really, for us, has become a game changer, because it really allows sort of screens at a different level. You know, we've done 100,000 cells in one day last week, and I really think that this will make a difference and will sort of change the way we conduct these experiments.
And I just wanted to mention that it's been fantastic to work with all of you. So thank you very much. And I also wanted to encourage all the listeners to please don't hesitate to get in touch with any of us in case you have any questions about single-cell sequencing, sequencing, or the crop-seq method. We're more than happy to assist.
Yeah, and a few final words from myself as well before I say thanks to all of you. Working for BD Biosciences with such an innovative surrounding and partnering with people like Tilmann, like Pawel here is really great fun. And we as BD we really want to support the biology and advance biology by bringing these innovative tools to market. And I'm very happy to see the use cases that are coming out of this, as we have just it has just been demonstrated by Tilmann. Thanks a lot.
Before we go, I'd like to thank the audience for joining us today and for their interesting questions. And once again, questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. We would like to once again thank our speakers for their time today and for their important research. We would also like to thank LabRoots and our sponsors Illumina and BD Biosciences for underwriting today's educational webcast. You can visit the webinar on demand. LabRoots will alert you via email when it's available for replay. That's all for now. Until next time, goodbye.
Bye.